So hi there. Good morning, everyone. James Jones, Research Coordinator at Boston Children's Hospital in the Computational Health Informatics Program, where we operate as one of the teaching hospitals for Harvard Medical School, and where I work very closely with Dr. Gil Alterovitz, who's one of the co-chairs of the HL7 Clinical Genomics Work Group. And due to a travel issue, I've been upgraded from a featured guest of this lecture to driving the discussion from the start. It should be good, though. I think we'll have a good time and get into kind of some details of one of the bulkier implementation guides out there for FHIR right now, and see some special use cases here in FHIR Genomics. So today we're going to be looking at the capabilities in FHIR to communicate genomics data, in particular starting with a little motivation and significance of some recent and upcoming work, and then we'll be going over the tools that are currently available and recommended to you by the HL7 work group, including the IG there, the implementation guide for genomics reporting. I'll be looking at the structure, in particular some of the decision of scope and design decisions on there, and looking in particular at the profiles. So we've got a lot of profiles that we define in there, and we'll be actually going through that kind of closely to show you where this discrete data should be being sent to some extent. And then we'll get some genomics report examples towards the end there, and then we'll get some questions in before we close. Hoping to run this a bit like a pop-up. If there's anyone who has any questions about particular slides or interesting kind of anecdotes, stuff like that, feel free to stop me and we'll see what we can do to get the information going. So real briefly, motivation and significance for genomics here. I mean, classically, the interest for using clinical standards for capturing this stuff is back to the Precision Medicine Initiative of the NIH. And we'll focus mainly on modernizing these genetic lab reports, which are archaic in a lot of places in the world. And then a lot of us mainly focus on integrating clinical and genomic data for point-of-care apps specifically. And lastly there, the last major motivation we have is for facilitating downstream research and analysis of these genetic results, which could be potentially very big in the future if we can get the infrastructure right. In particular, as whole genome sequencing gets cheaper and cheaper, it's more readily available to get this data, this wealth of data, and to get some extra use out of it rather than having to redo it every time you need a single test for something would be amazing. So there are some pretty legitimate concerns around quality of reusing this data sometimes if it wasn't initially looked at specifically and verified for specific variants, because a lot of these genes have regions or sub-regions that are hard to call. And if that wasn't done, then we got to be careful about that. But we want to make sure that we're enabling this as much as possible and not hindering or blocking its development. So real quick, for those who need it, the Precision Medicine Initiative's approach for looking at an individual's whole health data story, especially genetics, to determine their best care options. Major areas for improvement using this, including drug resistance predictions, transplant acceptance, and other major health concerns. One of the main focuses, like I said here, is modernizing these genomic lab reports, which are currently being generated by labs as PDFs. And a single report, which is sometimes printed out, signed, scanned back in, and emailed, or even faxed to the final destination. And the clear solution here is to use structured data to kind of convey this information, and FHIR, we believe, is the best approach for that for a number of reasons. In particular, there's no real standardization between labs or even between tests at the same lab sometimes, and it is difficult to read. I can't even pronounce some of the words on these labs that I'm looking at here. To look briefly at the apps that we're looking at here for point-of-care apps using the data. So a service request would be placed in the EMR under FHIR, and it would be received at the Genomics Lab, which then receives that in the specimen and goes and does the sequencing tests and returns the data back as much as they can in FHIR to the EMR, which then can send that to a Smart on FHIR app and which can also query any sort of genomic data or clinical data that's available. And there's an option here on the right to connect to external services and APIs, which can be incredibly useful for a number of broad use cases there. One of the other special things about this is it'll be scoped entirely through the EMR. So it'll be completely within the clinician's workflow and able to, already have the patient's information there with it. They don't have to take the information and then go externally and enter it into 5 or 6 different databases to see what they are looking at before they can generate their report. And of note, the Smart on FHIR specification here was developed out of Boston Children's a few years ago. And sort of applying it to these low-hanging genomic fruit was one of the first things that we did with it. Just to look briefly at those 2 apps that are mentioned there, the top one on the right is the Smart Cancer Navigator app, which will take a patient's cancer variations there and link them to external databases so the clinician can see one screen or a couple screens there, any clinical trial information or other kind of information about the gene and the pathway involved. And that was actually based off of recommendations of an ASCO workshop. And then the other app there is the Precision Cancer Medicine app, which was to show patient kind of variations versus a population from a statistical standpoint, how they're different or similar to others who have similar cancer stories. So for looking at the downstream research and analysis, it's been a vision that seems to quickly become more or less a reality or close to it. It's the notion of a JAX, I would say, not a GAX, unless you're one of those GIF people. But it's Genomics Archive and Communication System. So it would be much like a PACS for imaging data or pictures. You'd be storing the raw genomic data externally in a data store somewhere, and as long as it's able to communicate in FHIR when it's being queried, this is really interoperable for a lot of different use cases here, for either going to a Smart on FHIR app or for using CDS hooks directly into an EMR. And this was highlighted a bit in the Connectathons previously this year, and there's actually an article of that in the HL7 newsletter. You can read a little bit more to go a little bit more into detail there. This is a picture that was in the newsletter there of the diagram of the prototype that was carried out in San Antonio. So to describe this real quick, if you follow the numbers, so a medication is prescribed in the EMR here, which then triggers a CDS hooks event, and the folks at ELMU and Intermountain that we worked with had written some custom rules and translation and querying or retrieval algorithms to get data that was stored up here in VCF form, particularly from the 1,000 Genomes project, just de-identified information just for test purposes here, to translate that into FHIR diagnostic reports based off of any known drug-gene interactions for the drug that was being administered or started to be administered in the EMR. So if there would be, for instance, a CYP, was it 2C9 variation there, if you're sending a codeine or different drugs like that, it would say automatically, it would create the query, it would go and it would create the report and say, hey, there's a variant here that means that they're probably going to metabolize that specific way, so don't do that. And it would bring the information card right back. And what's really special about this is that the clinician doesn't even have to know anything about the genomic interactions here. As long as that's being coded, reliably in the rules engine based off of CPIC or ACMG guidelines or whatever is necessary there, they just go and say, okay, here's your information. It's up here. You did that. I don't need to look at it. I can just try to give you this medication. And oh yeah, it looks like we'll do something else because of the genes that we have just backed up in the JAX, which is great. And again, we're pretty far from being able to clinically use VCF results without special use cases in terms of the clinically kind of security here. Again, because of these regions that are difficult to call, and if that's not something that was completely verified, you don't know 100% that there may or may not be something else there. But it's a great start. And we'll look at the FHIR Genomics tools now. So the first stop should be the Clinical Genomics workgroup. We've got a Confluence page up there, pretty easy to find. We've got a Zulip chat or 2 or 3. We're happy to start more if people want to come in specific topics. We'll happy to break out and do that. And there's a listserv we're pretty active on. We've got 3 weekly conference calls online. It's Monday, Tuesday, and Thursday. They've got slightly different topics. So they're 11 o'clock Eastern time. Feel free to check out the work group at home on Confluence there. It's got more information on that. Monday are the FHIR calls specifically, where we look at things that are or concerns that are specific to FHIR and FHIR Genomics. Tuesday, we tend to do voting on the GForge issues, in particular for the implementation guide, which we'll go into in a bit, and other kind of concerns. We also have people who are experts in V2 and CDA and some stuff like that, but that's not super active in the group right now. And then Thursday is a bit more of an information modeling, looking forward towards kind of integrating this into maybe something more granular. And then the actual artifacts that we have here, we've got the domain analysis model for clinical genomics, which we'll get into some slides after the IG if we got time and people are interested, which goes through in a standards and platform agnostic way, looking at use cases in clinical genomics and workflows and kind of identifying the different stakeholders there and the different kind of opportunities you might have to develop apps or programs in different ways. You can kind of approach that with different languages. But the main focus is going to be the implementation guide for Genomics Reporting. One last thing here, Agile Genomics, the consortium for Agile Genomics is a new group inside the FHIR Foundation, trying to help start up groups who are looking to use genomics here in FHIR as to get them moving. You can check out that website there for more information. So yes, the implementation guide. How many of you have seen the Genomics Reporting Implementation Guide? Okay, that's about what I was expecting. It's a little hard to find. You can try Googling for it, FHIR Genomics Reporting. We might change the name to make it a bit more accessible, or make it more queryable, on SEO optimization, stuff like that. But you can find it also on the FHIR IG registry there on the left. And it's also linked through the ho7.org FHIR Genomics page, which is a bit interesting. So on R4 and above, you'll find a link there. There'll be a little STU note, much like this one, on the genomics page there in the core FHIR specification that will point you to this implementation guide. On R3, you will not see that note. You will just see what is deemed now our old guidance. Like I said, when Smart on FHIR first came out, we wanted to start using genomics right away on it. And so that was actually back in DSTU2. And there were some really rough, kind of just hacked in with extensions, basically. And you don't really want to rely on extensions too much because you have to specifically tell every server and client what extension you're using. And you have to decide these things in order to be able to communicate and not lose this information. So we've refactored a lot of that into this implementation guide, which is based on R4. But most of the design decisions and everything will translate back into DSTU2 and other things like that. And there are pages in the guide that talk about the technical kind of mappings that need to be worked through there. Great. So we'll go a little bit more in detail there. The kind of mission statement of the guide, which is not only slightly ambitious to cover all aspects of human genetic reporting. We've left actually a lot of room for more rigid guidance in several use cases, as you'll see. But we want to be able to report all classified and also unknown variants as much as we can, along with evidence of their known clinical significance. And we want to be able to scale that to the whole genome. So one of the notes is that we don't focus too much on workflow at this time. Really, the main focus is the underlying data structures, and then what guidance past that will come in the future. And this is a wall of text here. So it's intended to be the mother guide for genomics implementations. The US in particular will likely need a separate guide that will go adhere to ours, but also then import certain or spell out specific US specific requirements like CLIA certification issues and the use of SNOMED codes and things like that that are not universally available. We don't want our folks in Germany or anywhere else in the world to have to rely on these SNOMED codes because their countries might not necessarily have paid into the licensing for them and won't be able to do it. So to make it as adoptable as possible, these are some of the design decisions we've made. And another thing there, the big thing is to use observation and diagnostic report as much as we can. Observation is normative, yay. And to minimize the extensions, like I said. There's some interesting thoughts that have gone into this bit here, where genomic information can be conveyed at different levels of granularity. You can say there's a database of these certain variations. You can say just the code for that one variation. That might be enough sometimes to portray, okay, yeah, it's this known SNP in this database. Sometimes we need more granular information on that, particular variants of unknown significance, if we want to be able to reinterpret them later as more information becomes available. And then maximizing discoverability and queryability of the data is very important for downstream use as well. And yeah, I should briefly mention that there is also the V2 guide and the cytogenetic guide. Cytogenetics is in a very strange place right now in the IG. We took out most of the guidance there because it's not being done so much anymore, it seems. Most of the newer technologies are kind of overshadowing it, and it's a bit more of an edge case in a lot of the experts that we've been speaking with. So we'll be focusing more on that in the future. I don't know if I mentioned this, but this guide has passed 2 ballots already, and it's looking to be published by the end of the summer. So the draft is still currently available online. So looking at the table of contents there, we've got a number of pages, and the artifact list would go all the way down past the floor. And there's actually a little nav bar up top here with a couple drop-down menus, thanks to some people who mentioned that we should do that in the recent ballot. And that's actually really nice. Now you can kind of get around the guide pretty quickly to these major different pages that talk about the different use cases. And where it says FHIR spec right there lines up with the resources on the actual core FHIR spec, which is really useful, I think. Just a little Easter egg. Yeah, so going into if we want to go on where it would be here, the reporting overview section, or on the table of contents, general genomic reporting. There are a number of large diagrams and texts. This diagram here is talking about the main kind of build of the report, which is the Genomics Report, and the different observations that it carries under the results. We'll just kind of go through in this next section the different diagrams that are in the guide in particular, kind of how to read them, what they're kind of saying, and that'll give you the idea of the larger picture, which if you want to see it, that's the larger picture with all the different profiles that we have in the guide. Yeah, so we'll kind of build up to that in baby steps. We'll come back to this slide. So looking at the Genomics Report here, you notice that we've defined the category and the code. It's a LOINC code, so it's freely available. And we've added some extensions there for related artifacts, if you need to get citations, stuff like that. There's a spot for media attachments. If you need to send a VCF file, this is one way you can do that, or some other documents there. You can attach it as a binary, or you can send like a URL to a external repository, stuff like that. If you want to send the generated report as a PDF that you're still making in your lab, you can do that as well. There's been some guidance recently about reporting out or referencing to subreports. If, for instance, in your workflow, some sequence is being done at one lab and then extra verification or annotation is being done somewhere else, those might want to actually be different reports that are being referenced inside the report, as opposed to all being stacked onto one gigantic report. So there's extensions there that allow that. And then we'll get into the results, which are all observation profiles that we've defined in the guide. So starting kind of from the bottom up, we've got the region studied profile, which is able to show or to describe the region that was actually studied, as opposed to just where the result's coming from, because you don't know if you're not reporting something. Does that mean there was nothing found there, or that you didn't look there? That's a big concern that we have. So being able to specifically say, here's where we looked. Then you don't want to make the discussion that there's nothing found there unless it's being stated specifically, but it's important to have the information of that granularity of what regions you actually studied. And you can mention the kind of coverage there and stuff like that. And we're looking at expanding this in the future to get more granular quality assurance information. And then moving up, we've got the findings, which are the kind of actual genetic assertions of variants. It could be also named genotypes, stuff like that. Moving up from there, we've got the implications, which are the clinical relevance of these different findings. So it would mean like medication efficacy or pathogenicity that we've got there. And then there's an overall interpretation. And note that all of these can be arbitrarily grouped for representation using this grouper observation up there. There's a lot of variability in reports, like I had said, on the PDFs, and labs have different approaches that they want to take to these things. They might want to structure all of the pathogenic variants at the top and then some other information past that, or they might want to structure it by gene or by region studied. And we don't want to necessarily tell them they can't do that anymore, but you have to be careful just knowing that it might not be directly hanging off of one report. You might have to go through some iterations of observations to find where the results are. So yeah, again, one thing back here, some of these are red outlines. I don't know if hopefully you're not colorblind. I've been trying to get them to redo the structure of these so they're more colorblind friendly. But the outlines here are, if they're red, that says for diagram only. That just means that it's not one particular profile. It's actually representing a group of profiles that have a common set of properties. So for instance, like I said, finding, you're not going to make a genetic finding observation. What you're going to do is you're going to have all of these different variant, genotype, haplotype, structural genetic finding, or sequence phase relation. Those are the different findings that you can use, and they all fulfill the same purpose, the same slot there on the guide. So the implications here, you can see, have also split up a bit. There's different use cases here. Largely, there's pharmacogenomics. There's this sort of germline, inherited disease issues, and sort of cancer somatic issues as well. I'm not going to go too much into this page here, just to kind of give you a big overview. We want to go a bit deeper. So on the second diagram, on the general reporting page here, we've got some other reference options. The main point here is the fact that the implications are must be derived from a finding. You can't just say that they're pathogenic for something. Like, you have to say specifically what is causing that issue or that implication that you have clinically. And then there's also the overall interpretation, which will need to have preferably a finding. And then the recommended action is either a actual finding itself or one of the implications. And yeah, of note here, we're using derived from here for these observations, meaning that they're actually made based off of the other observations, as opposed to the has member, it's not brackets, has member, which is the relation between the grouper, which just kind of consumes them as a group of observations. Okay, and then we're looking on the next one, which shows you the other resources that are involved in genetic reporting. So it's unfortunately not all observation and service request or diagnostic report. There is a service request, there's a specimen that's involved, there's a patient, there's the recommended action, like I mentioned there, this organization or practitioner over there. And this diagram is actually going to simplify a little bit before it goes into publication. There's an open tracker or two to kind of resolve some of this stuff that's special cases that will make it a bit more simple. And then we've got supporting information. So one thing I mentioned was like citations for reports there, especially if you're talking about clinical guidelines for implications of things. We've also got family member history, which is taken into account for a lot of these things, especially if you're looking at inherited disease information. There might be someone, cousin or uncle or aunt or grandparent or something had some phenotype that we want to say might be a chance here to interpret the results differently, and also a risk assessment. And then some other clinical observations, particularly in the cancer space, if you want to see how treatments are holding up or what sort of phenotypes are happening there. All right, so here's where it gets fun. The findings, so this diagram you want to start at the top, the genetic finding. Like I said, this is not a single profile. This is a group of profiles that are underneath it here. If we're looking at the structural genetic finding, that's basically the only cytogenetic profile that we have currently. And that's, it's just a single like code on there. Basically, you just say what the ISCN string is, and that's the whole profile, more or less. And then there's another one down here, sequence phase relationship, which is interesting, as it points to two or more of these other findings and says if they're in phase or not. The zygosity there, if they're cis or trans. And you'll see there's a blue box here, molecular sequence. I've been told to tell you to be careful with this one. This is one of the parts of the old guidance. It is still used in the IG, obviously, here. And again, these diagrams are all currently available on, I think, all versions of the IG. The current one and the recent published ballot, not published. But the molecular sequence resource has some redundancies with the way that we're doing observations here, and it can be misleading if you are looking to describe variance with it, because it has a place where you can describe variance, but it's more of a way to describe a sequence based on the variance. You can say, it's like this sequence, but it has this variance. So be careful with that one. Really only use it if you need to absolutely send that granular level of data. Like a clinician is not going to care what the actual sequence in a region the patient has. That's not going to tell them anything. It's going to tell them, what the name of the variation is, or what the actual implications on the other sides of the things are. But there are some use cases that this level of data is necessary. And again, this wouldn't be a whole sequence. It wouldn't be a whole, here's a chromosome. It would be a chunk for a region, and you could point to other chunks as well if you need to. So building up from the left there, we've got the variant, which is the building block. And that's actually not a red profile anymore. It's a black profile. We actually had 3 profiles for variant, and we squished them all into one profile. That'll be fun on the next slide. But moving up to haplotypes. So haplotypes can be defined if you've got other variants. That's generally something that's inherited together. And there's a lot of special biological terms that qualify these things as being specific things versus other things. And then you've got genotypes, which can be defined from haplotypes or from variants themselves. And there's something else I wanted to say here. Oh yeah, so looking at the computable genetic finding, we're going to unpack this notation a little bit. It says component cytogenic location, component gene studied, and these are link codes. So it's got 3 components there, and up until now, all of the lines that have been on these things have been individual sort of lines of, or individual like FHIR data types. But these components are pretty complex things. So just zooming in there a little bit on, so this is the computable genetic finding, which again is not a real profile. It is a real profile, but it's not an implementable profile. It talks about these 3 components must be supported on these different profiles that are kind of inheriting this structure from it. These all have these common properties. So this observation here for the genetic finding abstract profile here is you've got the gene studied ID in this component, which you can see has this like system code. Has everyone seen this notation before? These tables, these differential tables for profiles? We've been going through the other diagrams and not the actual kind of standard. This is very, very key to implementation guides and profiles. Of note here, so this differential table here is only showing the differences between this profile and the base observation that it's derived from. And it's not actually going back to the base core observation, it's going to our other profile that we've defined, which is the genetic observation-based profile, which has a couple other components and things that we've defined on there. So you're only seeing how this profile is different from that profile on this page. You can look on the snapshot table if you want to see all of the fields and information there. But that page is awful to look at sometimes, especially you'll see variant. But these big red S flags, there's a lot of controversy over these. I know Mark Kramer in the back is really upset about all of the red S flags we have everywhere for must support, because we don't go into too much detail about what that means. And there's a bit of controversy in FHIR just in general on, do I really have to support all of these different things? And that's unfortunately not a question I'll be able to answer today. But we do want to say that these components that we've got here, you need to be able to say that there's a gene studied IG. which we want to have sent in this code. And the value for that is going to be a codable concept that uses HGNC notation. Likewise, for the next one, for the cytogenetic chromosome locations, we want to send that code in this component. So a server or generally a server, yeah, will know that that component is talking about the chromosome location. And it doesn't actually have a binding for that. So if you want to just text it out, that's what we're looking at here. And also the human reference sequence assembly version. But going even further into this, these codable concepts are not a single simple type either. You know, we've got one or more or codings, 0 or more codings, and potentially a text. So for each of those lines back there on those diagrams that was one component, you know, we've got these 2 codable concepts. You've got the code and then the value. So if we're looking at genotype here, to send the value for genotype, you could send it as text without any coding. So you could say it's A/C at SNP, RS, whatever that is. Or you can send, for a different one, we've got here encoded HLA terminology. We're sending the coding as this glstring.org. And we're giving a version of that because a lot of these systems do change. They get updated, in particular HLA does. And then the value for that is this code that means something in terms of that system. Have fun with that. And then the value for down here, if we're doing for pharmacogenomics, we might be using pharmvar.org instead, and the code value would be something separate there. And so the bindings for these different systems are mentioned down here, where the actual value for these components is coming from. Some of them are unbound. Like I said, you could just send text, or you could send whatever binding you like to send as a lab. We don't have a specific one there. Sometimes there's no specific code set or value set for these things, and sometimes there are multiple ones that you want to use, or very, very use case specific. And we're not saying that these have to be sent every time. You'll notice that the components here are 0 to star. It just means you have to be able to send the information there if the lab wants to send it. You want to be able to support receiving it. So here's Variant. Those are all components, except for the first top couple. I think there were 26 components on Variant. So potential components. 26 potential components on Variant. I don't think there's really a use case where you'd send all of them, but it is a huge Swiss Army knife of a profile. So you can send all of these different things. You can send the full location of the chromosome and what gene it's on, where the variant is specifically, what type of change it is, where the reference sequence is with different levels of granularity. You can send what the amino acid changes would be, the resulting thing, if it's a synonymous SNP, if it means that there's a changing in the acids there at the amino acid level. And also information on sort of presence in a sample or in like a population specifically as well. And it can even point to other variants to describe a complex change that's not covered by the haplotype or genotype use cases. So what we've got here, so the implications are next up. The implications require sending level of evidence, and they have related artifacts for supporting evidence there. And there's an interesting question here about secondary findings. So if you're doing a sequencing for a specific reason and you find something else that's very important that wasn't specifically asked for, under certain circumstances, the laboratory is obligated to send some of this information. Whether or not it gets to the patient or not is another question. But they have different standards depending where they are. So we have this Boolean, if you can see that it is unsolicited or not. You can say, oh, you weren't asking for this, but you might want to know that there's a very large concern. So the flagship profile here is the inherited disease pathogenicity, which has the associated phenotypes and the modes of inheritance, if it's autosomal recessive, stuff like that. There's a big question here, and we're looking for more guidance from FHIR here in the future. If these clinical implications should be done as observations, as how we've done it now. So they are patient and specimen specific. There's an opportunity for these to be definitional, for them to be based off of a certain knowledge base of these things, because these things tend to not change that much. At this point, what we're doing is we're generating, you're saying, hey, based off of this evidence that we see currently, X, Y, and Z, we think that this variant means this for the patient. People have already done that to some extent, and there's a possibility to, in the future, leverage other knowledge bases of these things to make it less or more streamlined, as opposed to being intricate for each observation there. So looking at these for use cases for pharmacogenomics, we're talking about a mandatory medication that we're talking about in particular. We're going to talk about, you can see if it's how effective it is overall versus how it gets metabolized. These are different profiles here that sit underneath the medication implication common properties there. Or if there's a high risk allele, that means that really don't give them that drug. It's not going to be good. And there are certain guidelines that specify how these different things are classified, where you can find that information. And there's also links up in here on the medication usage and recommended action. And this is one of the pages, if you go back to the table of contents. So we finished the reporting overview section, hooray. But there's still these other sort of specialized use cases here. We're in pharmacogenomics, and we're going to go to somatics as well. HLA, we're not going to cover it too much because we're running late. But so somatic implications are talking about a specific cancer. And we want to say, for that given cancer, how do the findings indicate the cancer? Or how do they simply or imply positive or negative outcomes for medication or therapies? It might not necessarily be a drug. It might be something other than drugs. And we're also looking at specifically how the medication might affect the tumor. So it's possible that a medication could affect the tumor positively, but not really change the outcome or the prognosis for the patient, which is difficult, but it's a distinction that needs to be made. So that is all the profiles. The IG, we made it. Does anybody have any questions? Want to stop me here before I get into something else? Yeah.
In looking at this, it looks very geared towards the whole treatment aspect. Company I work for, we do research stuff, so it's quite different in the flow. We bring in stuff from labs that are now less computers. We see them reporting not just PDF files, but structured data that we pull out. We pull out stuff from a lab potato DCF files, that kind of stuff. And it would be nice to be able to have that kind of stuff. And I can see the structure, there's support for that, but where you're doing the whole entire gene panels.
Yeah.
It's getting bigger and bigger with whole genome and all that stuff, right? Especially on the research side. Would that even fit in their infrastructure you were showing? It looked like it, so we just put one variant in there. Did I improve that?
Yeah, so the question there is, so in recent years, people have been getting somewhat structured data sent in these things. So from a research perspective, if you're sending more than one variant, stuff like that, is this still going to be suggested to be used for like a whole genome test, stuff like that? Is that what I?
Yeah, to support those other types of things. Because
Yeah.
And a lot of times when you're doing research too, you want to know not only did I find a variant, but did I not find variants, for the negative, that sort of thing.
Yep, exactly. And that's covered a little bit on the variance reporting page, which I don't have a major slide for because it's blocked out by the one variant profile. But yeah, the region studied profile gives you the opportunity to say, here's specifically where I looked. And we've got some components on there that you can break down subregions and say that these are different areas that didn't have anything, or these are areas that did have something. And you can point out to these things as well. These reports, if you're doing it in that structure, will get very large. If you want to send them all as one bundle, it can happen. It happens. We've done some with it. In particular, yeah, using the de-identified 1,000 genomes VCFs for that project there would be several hundred genes on these things. And it's possible to get through it with.
We've got customers like that have, they're more on the research side. They have hundreds of thousands of variants per patient. They do the whole.
Yeah, so the idea is you're saying patients or may have hundreds or thousands of variants that are detected in a sequencing panel, and whether you'd be presenting all of those variants necessarily in a variant class. And the answer is I would say no. You'd only be reporting the clinically relevant ones here. You'd have the capability to report any of them on demand as needed. But as long as you had the ability to take, we're looking at getting up server operations with this, so you can say, okay, I'm querying for this region. of this gene for any variants here. And you'd be able to take that, and your server would have to be able to respond back with a FHIR Genomics report saying, yes, there's something there, it's this. Or yes, there's 1,000, or not 1,000, hopefully you have a smaller query than that, if you can, at a time. But there's any number of findings that are there, and here they are. So you don't have to send all of the information in FHIR at one time, but you should be able to send any of it as needed. Does that make sense?
And the research you want to send them along because you're talking here about stuff that's very much well known. But if you're not in research, you don't know, and those things do change a lot. Not today, they may discover tomorrow it is.
Right, they do change a lot. The determination of what is pathogenic and stuff like that can change a lot. And so you would want to not necessarily send every single variant there, or you can send a way to access it, at least, with metadata. Yeah, yeah. In the sequence resource, there's a spot for repositories, which you can point to specifically, those things like that. And we can talk more, if you like, at the Meet and Code session or other stuff like that. There are a number of projects that are looking at doing this for sure. And yeah, there are some concerns there because observation is a bit bloated for sending thousands of genes. Yeah. Yeah, so there's a lot of overlap in our work group with GA4GH. A number of the co-chairs are pretty well involved there. So we know what's going on there and what's going on. And we're trying to get to some common ground. Absolutely, that's the goal, obviously, for interoperability and for genomics and health. Yes, yeah. So they are really research focused, and we're trying to do everything we can to make sure that that is still able to be represented clinically as needed. But we want to make sure that they're evolving in cohesively, you know. Rain?
And you also have a build section, or is it Meet and Code?
Meet and Code, yeah. Come on down. The special.
Will you be there?
Special topics table, I'll be there. I think I want to see Mark's talk later today, but other than that, I'll probably be over there. All right, how much time do we have left? None? That's it? All right, well, I'll skip through the actual report if you want to see. This is just 3 genes and a couple of therapy matches, and it ends up being 1,000 lines of XML. But HSBC takes it fine. You can just put it in the sandbox, and it's up there. You can query through it. You can find all these genes and information there. You could send, as much as that at a time with no issue at all. You could probably send even more of it if there's something that's more scaled up for that sort of thing. We can play around with that if you are interested. All right. Thank you.